Zachary
Thank
Prensky
you.
from LB Pharmaceuticals.
Thank you very much, and thank you for coming here today. I appreciate your time. LB Pharmaceuticals, a company that I co-founded 3 years ago, has a very unique business model. What we do is we go out and search for successful CNS treatments that are already either standard of care or in use in very significant numbers of countries overseas, but for whatever regulatory or financial reason have never been brought to the United States. That's the business model of what we're doing, and our first drug that we've targeted is an extremely successful drug in the area of schizophrenia. Now, if you're not familiar, schizophrenia is a debilitating illness that affects at least 1% of the United States market. There's 14 drugs that are approved in the U.S., a couple are branded, most are generic, and what people don't realize is if you fail those drugs that are currently on the market, while in Europe they may put you on AMISULPRIDE, in the United States we really have nothing other than clozapine. So there's an entire class of drugs that is used in Europe to treat patients that fail the existing first and second stage drugs. They're called benzamides, the best of which is being AMISULPRIDE. This drug is not available in the United States. All patents have expired. It was originally designed by Sanofi, and our approach has been to slightly change the chemical composition to improve the blood-brain barrier permeability, file patents, composition of matter, use patents, and so on and so forth, and therefore that gives us the financial ability to afford the extremely expensive development that any antipsychotic goes through here in the United States, a full phase 1, phase 2, and phase 3. If we get to market, there are 60 million prescriptions a year, and as you'll see shortly, AMISULPRIDE has an enormous market share in Europe, and we expect it to have a similarly strong market share in the United States for those patients who are currently untreated. Our main shareholder, besides myself and my family, is the world's largest generic manufacturer of AMISULPRIDE. Pharm, based in Switzerland, has invested almost $1 million to date, and we're currently completing our IND package and hope to be in the clinic next year. Here's a bit about us. All of us have extensive biotech experience. A number of my partners have been involved in more than their fair share of orphan and CNS drugs. We have an advisory board that's based upon some of the best minds in psychiatric studies today. Dr. John Kane, Dr. Glick, Christoph Carell, together have run more phase 2 and phase 3 studies in this space than virtually anybody else. So what have we done with this molecule? AMISULPRIDE has been around for 30 years, and like all approved atypical antipsychotics, it must be active at the dopamine, the D2 and the D3. It's also a very strong 5-HT7 antagonist, which probably is the reason for its strong use for cognitive impaired patients. Again, been around for 30 years. What we did is we took one of the hydrogens coming off the aniline nitrogen and we replaced it with a methyl group. In essence, a prodrug approach. We then ran a number of in vitro and in vivo studies to make sure that we haven't impacted AMISULPRIDE's unique CNS binding profile, but have improved its ability to cross the blood-brain barrier. Having done that, we looked at the toxicology profile, and all of this looks very positive, which is why we're now looking to move into the clinic. Now, again, a bit about AMISULPRIDE. This is one of the most cited papers in all of psychiatry. It is a meta-analysis published in 2013 in Lancet by one of our KOLs, Dr. Stefan Loy. You take a look at this on the top left. This is an analysis of over 43,000 patients in 20 years, the effectiveness of an approved antipsychotic to treat schizophrenia. There is nothing more effective that we have today other than clozapine and AMISULPRIDE. Now, once you give a patient that drug, what you need to know is how long they stay on it. Most patients who have a psychiatric disorder can go on and off drugs numerous times a year. So this chart over here shows you something very interesting. Of all these approved drugs, it has one of the lowest discontinuation rates. So that, coupled with its efficacy, gives you a strong understanding of the safety. In other words, if you are a European patient put on AMISULPRIDE, after 12 months, you have more than a 2/3 chance of still being on AMISULPRIDE. Once we get you on this drug, you stay on this drug. Of course, in the United States, we don't have that option. Here are the numbers. The bar chart's quarterly prescriptions. It's over 2 million annual prescriptions for AMISULPRIDE. It has a 2% market share of all CNS indications, not just schizophrenia. So if you included everything, sleep, depressions, what have you, even though it's not used for those, it has 2% of the entire CNS market. SULPRIDE, which is also a benzamide, has a 2% market share. So what you can say in Europe is that 1 out of 25 prescriptions written for any CNS indication is of a benzamide class. In terms of percentage within schizophrenia, it varies from between 5% to over 10%, such as countries like the UK. Why is it so different from other drugs that we have? It's as effective as a risperidone or olanzapine, but unlike those drugs, it has zero weight gain. Now, olanzapine is the only thing we currently have that's effective to treat negative symptoms. AMISULPRIDE also seems to have that ability to effectively treat negative symptom schizophrenic patients. In a recent paper, there was an analysis done compared to olanzapine and risperidone. Patients on AMISULPRIDE have lower risk of self-harm. And when a patient fails risperidone or olanzapine, either for safety or efficacy, more patients, over 50%, have had improvement switching from olanzapine to AMISULPRIDE. PANSS is the gold standard for analyzing how much psychosis a patient is undergoing. The higher your number, the worse off you are. So when you look at how well a drug runs in this area, you look at a decrease in PANSS. And for more than half the patients who switched to AMISULPRIDE after failing those two drugs, they succeeded. In a recent meta-analysis, just a couple of months old, AMISULPRIDE was the only antipsychotic approved, which includes olanzapine and risperidone, that outperformed the placebo in the treatment of negative symptoms. So despite all this data, AMISULPRIDE, totally unavailable here in the United States. We expect to change that. Now, what have we done to this drug? If you look at the far left where the arrow is, you'll see that's AMISULPRIDE. Of these 30 psychiatric medications, AMISULPRIDE is the least able to passively diffuse across the blood-brain barrier. A drug needs to get into the brain in order to work to treat a psychiatric disorder. So in Europe, there are many patients who take 600, 700, 800 milligrams of drug a day of AMISULPRIDE in order to be effective because so little of it crosses the blood-brain barrier. Now, in other cases, pharma companies, if you look at the, I don't know if this pointer's working here. Is it, maybe it's not.
Turn it around.
Turn it around? Oh,
yep.
So there you go. Okay. So thank you. So if you look at these two examples that I've given you here, these drugs only differ by a single methyl group. Here's proof positive that swapping out a methyl group at a key nitrogen on that molecule will increase its ability to cross the blood-brain barrier without impacting the efficacy or its CNS binding whatsoever. Okay. So here is AMISULPRIDE on the left, LB-102 on the right, the only difference being the stripping of one of these hydrogens off and replacing it with a methyl group. We haven't done much to change the molecular weight, but we certainly have been able to change its lipophilicity, and I'll show you that in the next slide. We filed patents a short while ago, and we have data showing that we believe that we have greater membrane permeability, and in a number of animal models, we ran it head-to-head against the AMISULPRIDE. We've shown as good or better efficacy in those models. So here's the first assay we ran. This is a straight Pampa assay. It's a membrane. You count 30, 60 minutes, see how much has crossed it. We were 200 times more permeable than AMISULPRIDE. This was a very nice result. Next step was to see, okay, we know we're more permeable, but has our methyl addition changed any of the key CNS properties? Well, no, they haven't. We're still a very strong binder at the D2, D3. We're still a potent 5-HT7 inhibitor, and we haven't changed the non-activity at any of these tox areas that would cause sedation, weight gain, anxiety, or what have you. So in other words, we have almost exactly the same CNS profile as AMISULPRIDE with the addition of this vastly improved BBB permeability. The next step was to look at the pharmacokinetics of our drug. The curves overlay almost perfectly, so we feel very strongly based upon this we should be able to formulate an oral dose formulation for humans almost exactly the way that AMISULPRIDE is dosed. The next step was to look at the receptor occupancy for specific dopamine D2 and D3 receptors. Now, as I said earlier, all approved drugs in this class must be D2 or D3 antagonists. Dopamine RO is highly correlated to improvements in PANSS. We took a look at our drug over here compared to AMISULPRIDE. Very, very, very nice results here. We can see a clear improvement at the 30 mg per kg dose, and this just missed being stat sig. So we see as good or better dopamine receptor occupancy in LB-102 versus AMISULPRIDE. The next step we took was to look at 3 different efficacy models of schizophrenia. The first one being the novel object recognition study. This is a well-published and validated model of cognitive impairment, so it leans towards patients that express negative symptoms. We were as effective as AMISULPRIDE in restoring cognitive function to these rats. The next study was the locomotor activity study, the LMA. Actually, we had a great firm up in Sweden. Charles River has a small lab there that just focuses on these kind of models. We're very happy to say that in this one, in the LMA model, we actually showed statistical significance against AMISULPRIDE in improving an amphetamine-induced rat. So here we show real superiority at the 30 mg per kg dose as compared to AMISULPRIDE. LMA is more indicated towards positive symptoms of schizophrenia, as is the last model, the mouse apomorphine-induced climbing study, where we showed improvement comparable to AMISULPRIDE. So what have we done after that is obviously look at the toxicity. You have efficacy, have you changed the tox profile? So far we've gone out 14 days, and we're very happy to say that based upon what we've seen so far, the MTD of 200 mg per kg is absolutely consistent with the published data on AMISULPRIDE, which is in excess of 7 times that 30 mg per kg dose where we saw significant efficacy. So we have a very nice range between where the dose is effective to where it becomes toxic, and what's most important is it overlays precisely upon the published data with AMISULPRIDE. We also ran a test for EPS, the bar balance test. We were also indistinguishable from AMISULPRIDE. So if you put that all together, what we know to date is LB-102 has a PK profile that's indistinguishable from AMISULPRIDE. We either have equivalent or superior dopamine receptor occupancy. We've run 3 different well-validated models, 2 in rats, 1 in mice, 2 in positive, 1 in negative. In the hyperactivity, we showed statistical superiority. In the cognitive and stereotypy, we were equivalent. And then in the MTD study, we showed equivalency in terms of tox with at least a 7 times coverage as compared to efficacy. Okay, what's next? A couple of weeks ago, we met with the FDA, and we've got some very good guidance in writing for them as to what our clinical program should look like. We should be complete with the IND-enabling studies by the end of the year. The first phase 1 could be started as early as April of next year, and based upon the written guidance we have, we only need between 30 and 40 healthy volunteers before we can then move straight into a phase 2A proof of concept study in acute schizophrenia patients. This would be a 1-week, 2-week single ascending multiple dose, start to finish, possibly 3 months. I know it looks aggressive. We've actually had a number of CROs take a look at this. We have bids on this. We hope to move very quickly once our IND is active. The phase 2A would really be capital constraint. We could do a 40, 60, 80 patient study. You're really looking for a proof of concept. You need to do acute. It has to be inpatient. 4 weeks is probably where you want to go out before you start to see significant separation of the curve. If you're looking for stat sig, if you want to save money, maybe you can run it a little shorter. The data from this is what we would use. Yeah, the data from this is what we would use to then go out and do a much, much larger equity round to fund the phase 3 program. So in summary, we have a novel antipsychotic that's very closely related to AMISULPRIDE with patents filed both in the US and worldwide. We're looking at the CNS receptor binding. It matches AMISULPRIDE. We looked at the PK profile, matches AMISULPRIDE. We looked at the dopamine receptor occupancy. It's either as good or better. And in 3 models, we showed the same or better efficacy compared to AMISULPRIDE. Our IND-enabling package should be complete by the end of this year. So in every measure to date, we're good or better than AMISULPRIDE. Now, if you take a look at the most conservative figure of the 2% that I showed you before and just focus on the schizophrenia market, this is a $1 billion drug. It's a $1 billion drug because patients don't have this option here in the United States. I think we can do better than that, but you know, in my 25 years of investing in life science, this is this is the best risk-reward profiles of any clinical program I've ever seen, and that's really why I founded the company to do this. We're currently in a process of closing our Series A, and again, we hope to be in the clinic next year. I'm happy to take any questions.
Yeah, on the on your length of trials here, doesn't the FDA speed this up? Won't they take any of the data that you have from the European trials?
It's an excellent question. Remember that our molecule is an NCE.
Right.
We've stripped the hydrogen and put on a methyl
Right.
group. So no, they won't accept that data. Number one. Number two, those trials were run in the 90s against first-generation atypicals. Actually, the bar is even lower than you think. The FDA's guidance is to run against placebo. So we're running against placebo. I don't even have to go up against standard of care. And there's a very good reason why. Because the patient that might respond to olanzapine is not necessarily our customer. So they just want to know that we're effective, and they believe that the market will then shake it out, and patients who fail those other safe drugs will then come to us. So the bar for approval is very, very low in this class. You must run a total database of 1,500 patients, however. So that's why this is expensive. Yes, any other questions?
What's your financing requirements and needs?
So we are currently in the process of closing a $7 million round, which should get us through our phase 1. The IND is the most expensive. In the FDA guidance that we've received, we have to do GLP tox for 14 days. We'd like to exceed that. We think the market wants to know the safety here is impeccable. So we plan to go out 30 days on our tox studies. That's a big chunk of the cost. The phase 1, 20 to 30 patients, is $1 million, $1.5 million, and from there we can go straight to a phase 2. So my guess is it's about $7 million, and then at the end of 2019, we'll look to raise another 10 to 20 million. There are 2 other drugs that are similar in how they're positioned worldwide in the CNS space. They are standard of care for orphan illnesses. They're imported by approximately 10 to 20% of the patients affected by them in the United States, and the remaining 80% just die. So we have some other products that we'd like to go after in the CNS space, where you can put up another, $1 to $2 million in proof of concept, create some IP, and do that. So maybe you have a $10 million in total. We hope to have this wrapped up in the next, 8 to 12 weeks in terms of getting that financing in. Last questions?
We have one more minute for questions. Please ask a question, because otherwise I'm going to ask him to describe this rat schizophrenic model.
Oh.
I read that, I'm like, wait, models of rats with schizophrenia.
It's great stuff.
But please, if someone's got a more serious question, please go right ahead.
Nobody.
All
It's
right,
actually
you're stuck.
great.
There we go.
No, it's great. So what they do is there's 2 models. They give them PCP, which makes the rats go crazy. Okay, so you give the rat the PCP, they can't tell the difference between something they saw 2 minutes ago and a novel object. They're totally confused. And then the other way you do it is you give them apomorphine. Same story. They'll just go crazy. They'll run around like nuts. They can't move this arm with this leg. They can't get that together. So yeah, you look for rats where you give apomorphine, PCP, and then you try to quote unquote calm them down with your medication by restoring them to normal function. It's fascinating. Yes.